cholesterol metabolism. The topics included in this lecture are cholesterol metabolism, regulation, as well as hypercholesterolemia. What is cholesterol? Cholesterol is a white waxy substance. It's an alcohol with an OH group attached to carbon number 3. Carbon number 3, or the OH at carbon number 3, is the site of acidification with free fatty acids when cholesterol ester is formed. The sources of hepatic cholesterol. After lipid digestion, chylomicrons and other lipids are absorbed and bypass the liver to the circulation. The chylomicrons are processed in the circulation where they are converted into chylomicron remnants that are delivered to the liver, and this represents the first source of hepatic cholesterol. The second source is the reverse cholesterol transport via the high-dense lipoproteins, which take up cholesterol from tissues to the liver. High-dense lipoproteins take cholesterol from the chylomicron remnants and intermediate density lipoproteins. A third source for hepatic cholesterol is the de novo synthesis from the precursor substrate acetyl coenzyme A. So what about the fate of hepatic cholesterol? The very low density lipoprotein is synthesized in the liver from chylomicron remnants and is delivered to circulation where it is converted to the low density lipoproteins after binding to cholesterol and the apoprotein P100, which all form the transport vehicle of cholesterol to tissues. Part of cholesterol is excreted with bile to the gastrointestinal tract, and this represents a means of reducing the cholesterol level in blood. If bile becomes over or supersaturated with cholesterol, it may precipitate, and if infection occurs, this may lead to gallstone formation. This is all beside the sense of bile from cholesterol. So, these represent the fates of hepatic cholesterol. The biosynthesis of cholesterol, or de novo synthesis of cholesterol, starts with the substrate acetyl coenzyme A, or the active acetate. Active acetate comes from the oxidative decarboxylation of pyruvate, which is a result of glucose oxidation or glycolysis process. So pyruvate is oxidatively decarboxylated by the action of pyruvate dehydrogenase. Another source of acetyl coenzyme A is from the beta oxidation of fatty acids. So these acetyl coenzyme A molecules are delivered to the liver, where the cholesterol synthesis starts. Two molecules of acetyl coenzyme A condense by the action of a thiolase enzyme, forming acetoacetyl coenzyme A. And this occurs in the liver placenta, intestine, skin, and suprarenal glands, which are the major sites synthesizing and utilizing cholesterol. The second step in cholesterol synthesis is the condensation of a third molecule of acetyl coenzyme A with the previously formed acetoacetyl coenzyme A molecule by an enzyme which is hydroxymethylglutarate coenzyme A synthase, followed by the rate-limiting step catalyzed by hydroxymethylglutarate coenzyme A reductase with the formation of mevalonate. This last step is the rate-limiting reaction of cholesterol biosynthesis, where pharmacological intervention can occur by statins that inhibit hydroxymethylglutarate CoA reductase and serve as a cholesterol-lowering drug. Mevalonate undergoes several reactions forming anosterol, sinosterol, desmosterol, and finally cholesterol. Talking about the regulation of cholesterol synthesis, regulation of the levels of cholesterol in the circulation as well as in the tissues occurs at different levels. The regulation of synthesis and uptake. As the cellular content of cholesterol increases, it exerts a sort of feedback regulation where the elevated cellular cholesterol itself inhibits the sense of the regulatory or key regulatory enzyme, the hydroxymethylglutarate CoA reductase. Also, there is reduction of low-dense lipoprotein receptor synthesis, and these two processes contribute to subsequent reduction of cholesterol uptake. A third effect is activation of an enzyme, acylcholesterol acyltransferase, which is concerned with cholesterol storage, whereas cholesterol is acetylated or esterified and stored in the form of cholesterol ester. Another level of regulation is exerted as a transcription level of hydroxymethylglutarate CoA reductase gene. And as mentioned earlier, cellular cholesterol is a major effector of this process. The transcription of hydroxymethylglutarate CoA reductase gene is regulated by a protein SREP or steroid regulatory element binding protein, which is synthesized and activated in the endoplasmic reticulum and Golgi, then enters the nucleus, where it binds to steroid response element on the DNA or the gene promoter, where transcription starts. And as shown in the figure, elevated cellular cholesterol negatively affects the SREP, where SREP is bound to another regulatory protein, which is SCAP or SREP cleavage activating protein, leading to cleavage of SREP with the reduction of hydroxymethylglutarate CoA gene expression, or the HMG COA Reductase gene expression. Back to the regulation of cholesterol synthesis, we have the hormonal regulation. After meals, insulin induces activation of hydroxymethylglutarate CoA reductase by activating a protein phosphatase that dephosphorylates the hydroxymethylglutarate CoA reductase with subsequent activation and increased cholesterol synthesis. So, as we see, feeding promotes cholesterol synthesis by providing the substrates for cholesterol synthesis, which are acetyl coenzyme A and the reduced NAD phosphate, and activation of the regulatory enzyme hydroxymethylglutarate CoA reductase. During fasting and the periods of food deprivation, we have glucagon, the hormone that activates adenyl cyclase with increasing levels of the cyclic AMP and subsequent activation of a protein kinase that phosphorylates the regulatory enzyme hydroxymethylglutarate CoA reductase and inhibits its activity. Subsequently, cholesterol synthesis is reduced. This process is AMP dependent, so when ATP is reduced, there will be no need for cholesterol synthesis. So, as we see that fasting is a good cholesterol lowering strategy where the hydroxymethylglutarate CoA reductase activity is reduced. So, as you see, the covalent modification of the enzyme is key for regulating cholesterol synthesis. The hydroxymethylglutarate CoA reductase is covalently modified by phosphorylation-dephosphorylation reaction. The active form is a dephosphorylated form. This is achieved by a protein phosphatase that is activated by insulin, whereas it is inactivated by phosphorylation by a protein kinase, which is activated by the hormone glucagon during fasting and starvation. And hence, this explains the role of fasting in down-regulating enzyme activity and cholesterol level reduction. To summarize the previous regulatory mechanisms for the activity of hydroxymethylglutarate CoA reductase, we have short-term regulation by phosphorylation, dephosphorylation, and long-term regulation by proteolysis of the enzyme itself and cholesterol-stimulated degradation of the enzyme. Also, gene expression and regulation by the steroid sensing domain and the strip protein. So, if we understood the previous points, we can now explain how does fasting inhibit cholesterol synthesis and explain how the feeding fasting cycle regulates the cholesterol metabolism. And one more question before the previous two questions. We can explain how carbohydrate-rich diet leads to cholesterol synthesis and hypercholesterolemia. So, if cholesterol synthesis is dysregulated, hypercholesterolemia may occur and patients need to reduce their blood cholesterol levels pharmacologically by drugs. And as we know that bile acids, a great percent of bile acids is reabsorbed and recycled in the liver, where they have a great percent of the cholesterol with them, and only small percent passes with feces. So pharmacological intervention will block the recycling of bile acids and consequently forces the liver cells to utilize more cholesterol in bile acid synthesis, and this will share or contribute to the lowering of cholesterol levels in blood. Examples of these bile sequestering agents are cholesterol and cholestyramine, which are anion exchange resins, and as mentioned earlier, they act by blocking the intrahepatic recycling of bile acids and thus forcing the liver to utilize more cholesterol in sense of bile acids, and this will contribute to lowering of blood cholesterol levels. The bile acid sequestering agents are used in combination with other drugs, which are the statins, and statins are hydroxymethylglutarate CoA reductase inhibitors. They reduce activity of the reductase, which is a regulatory enzyme for cholesterol synthesis, and this gives better results in treatment. As mentioned earlier, if dysregulation of cholesterol by synthesis occurs, hypercholesterolemia may occur. An elevated cholesterol level in plasma above 220 milligrams per deciliter is considered as hypercholesterolemia, where the normal range lies between 150 to 220 milligrams per deciliter, and hypercholesterolemia, as we said, results from dysregulation of the mechanisms of cholesterol synthesis. This disorder may be familial or secondary to a primary disorder. The familial type may be due to a deficiency of the low-dense lipoprotein receptor, and this is a genetic defect of the low-dense lipoprotein receptor gene. And as we know, the low-dense lipoprotein receptor is responsible for internalization and uptake of the low-dense lipoprotein with its cargo of the high cholesterol content. Other causes or secondary causes of hypercholesterolemia include a carbohydrate-rich diet, which provides the substrates for cholesterol synthesis. Carbohydrate provides acetyl coenzyme A and reduced NAD phosphate for the synthesis of cholesterol. A second cause is diabetes mellitus, where the lipid and all other types of metabolism are disturbed. The low-dense lipoprotein receptor and its metabolism are deranged in diabetics, and there is decreased clearance of the plasma lipoproteins, especially the low-dense lipoprotein with its high content of cholesterol. A third cause is hypothyroidism, And it is known that thyroid hormones stimulate bile acid synthesis from cholesterol. So these cases are associated with hypercholesterolemia and maybe gallstone formation. Other causes maybe some types of obstructive jaundice, which leads to decreased cholesterol excretion through bile ducts with bile acids. Also, obesity and nephrotic syndrome cases are associated with hypercholesterolemia, but the causes are unknown. Clinically, hypercholesterolemia may be manifested by hypertension, atherosclerosis, or atherosclerotic problems presented by myocardial ischemia. Other presentations may be with cases of hypothyroidism and their complications, skin manifestations as xanthomas of the eyelids, or eruptive xanthomas associated with hypertriglyceridemia. Also, it may be detected by routine laboratory investigations. Another problem that may be associated with deranged lipid and cholesterol metabolism is the fatty liver. The causes are summarized as overmobilization of fat and lipids to the liver, undermobilization of fat from the liver, or excessive carbohydrate intake, which leads to excessive lipogenesis and further accumulation of fats in the liver. This condition requires treatment. Treatment of the condition of fatty liver is in the form of agents that help mobilization of lipids out of the liver, and these are called lipotropic factors. And these factors are essential for the phospholipid synthesis, which are integral components of the lipoproteins responsible for mobilization of lipids and cholesterol and their transport across the circulation and tissues. These agents are in the form of choline and inositol. Some of these agents are provided by essential amino acids, and they are important for synthesis of choline, carnitine, and apoprotein synthesis. Also, essential amino acids as methionine and serine, and vitamin B12 and folic acid, which are important for the transmethylation reactions. How to treat hypercholesterolemia? The first line of treatment of cases of hypercholesterolemia are the lifestyle changes and dietary regulation. Also, pharmacological treatment by drugs as statins, which are hydroxymethyl CoA reductase inhibitors. These drugs are structural analogs of the hydroxymethyl CoA, which is a substrate for the enzyme, and thus inhibit cholesterol synthesis and reduce intracellular cholesterol. This will consequently lead to low-density lipoprotein receptor synthesis upregulation and increased uptake of cholesterol. And this will finally lead to lowering blood cholesterol level as we reduce intercellular synthesis and increase uptake from the circulation. Another agent is niacin, or nicotinic acid, which has a minor role in treatment of hypercholesterolemia due to its side effects and complications. Also, we have the oat bran, which are the soluble fibers of oat. These fibers bind bile acids with their cholesterol content, and this shares in lowering cholesterol blood levels. Other drugs used, as mentioned earlier, in combination with the statins are the bile acid sequestrants or bile sequestering agents, and their examples are cholestyramine, cholesterol, which sequester bile acids in the gastrointestinal tract and block their recycling. But these drugs also block absorption of fat-soluble vitamins. They may also bind some polar drugs as warfarin, digoxin, and thyroxine. So they have many side effects and drawbacks. So the best strategy for treatment will be the soluble fibers and the statins, as well as lifestyle change and dietary regulation. The last topic in this lecture is hypocholesterolemia, which is decreased level of cholesterol in blood below the normal level, which is 150 as a lower limit, 150 milligrams per deciliter. Some of the causes of hypocholesterolemia. First is hyperthyroidism, which is a common cause for hypocholesterolemia. Other causes may be pernicious anemia, hemolytic jaundice, malabsorption syndrome, and other causes that will be discussed in a separate lecture. Thank you. You can find other relevant information about this lecture and other lectures at this site.